It's my pleasure to introduce our second speaker, Professor Swapnil Rane, the Director of Digital Pathology at the Department of Pathology, Tata Memorial Hospital in India. He will be presenting on implementation strategies and challenges in image biobanking. Please.
Thank you very much, Professor Chan, for the introduction. And I would like to thank the organizers to have me here for this particular presentation, share my ideas on a topic which is close to my heart. I would also like to thank for the effort taken by the organizers in setting up the Asian Society of Digital Pathology. And it is required to have more regional organizations which are concerned with actually the adoption of digital technology in pathology and related domains, and to train more and more people in this particular area, which is still in infancy in many of the countries that we service. So I'm going to start. The topic assigned to me today is about implementation strategies and challenges in image biobanking. And this actually comes from our implementation of image biobanking that we have done. It's a multi-institutional effort that we have done at Tata Memorial Center. We have collaborators from AIMS, PGI, Chandigarh, and some other centers from India. And we are now at a critical stage, and we are actually looking to expand the effort to include more and more organizations. I will talk about our reasons for going this way, and what problems we faced and how we tackled some of them, and how we are trying to deal with all of this. Most of my work is funded through Government of India fundings from different organizations, Department of Biotechnology, my own parent organization, Tata Memorial Center, and Department of Atomic Energy. But we also get some other sub-awards and other funds from various other international collaboration that we have within US, UK, the rest of the Europe as well. Whatever work I'm speaking of is at various stages of development, and as the things progress, at this point I'm going to give a flavor of where we are going and how this resonates with the rest of the digital world. While I speak, there's a big team of people who have supported us during this entire endeavor, and it is not without them that it would have been possible to do any of the work that we have been doing and will do in the future. The photograph actually shows a team of pathologists and computational pathologists and technicians who actually work together to build the bank, as well as do most of the algorithm building that I'm going to talk about over here. So this is broadly what I will speak, why we are talking of image banking, what kind of data are we talking of, what data should be there, should there be only images, should there be some other information, what data should be there, and the other issues that we face, including the regulatory and policy issues during the course and during the entire life cycle of an image bank. So the digital universe has transformed the way we do everything. We have gone from analog to digital in most of the fields of our lives. The healthcare sector is no exception. A lot of the data that is driving the engine of the innovation in cancer care, the way innovation is progressing in cancer care, is being driven by a lot of data becoming available at the fingertips of many researchers, as well as the scientists and medical personnel who look at this data and interpret and make hypotheses and actually test those hypotheses in real world life. They further drive a lot of the clinical trials that take place across the world, and which eventually comes back to the hospitals. In a number of ways, the image banks and data banks actually work like biobanks, except that there are no physical tissues, but there are digital images as well as data. And there are a lot of similarities between the two pathways, and in many positions, it is possible that both these things coexist in a single bank. Even if not, they can actually talk with each other and create a lot of interaction between the data as well as tissue banks, and drive this engine of innovation in cancer care. As more and more data actually comes from the real world application, real world hospitals, this is going to become more and more generalizable. Currently, most of the data comes from silos, and many of the AI algorithms suffer from reduced generalizability across unseen data populations. So that was one of the major major motivation behind actually going forward with this image bank in India. We had tried using the TCGA data sets and the other data sets available worldwide, and we started applying and testing them on our patients. We found massive drop in performance due to a number of reasons, including biological as well as ethnic variation, and obviously the variation in data that comes from the lab-to-lab variation and the differences in the processing quality of the tissues as well as the capturing of the images. So that was our biggest motivation, and then we realized that this problem is obviously something not that we face alone. There must be many people. There must be a lot of teams and people who face this particular problem, and we therefore decided that yes, while we go about doing our AI work, our image banking, building algorithms that work on our patients, and try starting deploying them in real life, we would make this data. We would like to make this data available to the rest of the researchers outside our particular center. So we set out with essentially not just collecting and collating and organizing the information, but also put in a lot of effort in anonymizing without losing a lot of the linkages between the data fields. Sanitize, make sure that this is representative of the population that we are serving. And at the same time, we wanted to make sure that the data is used reasonably and scientifically and ethically. And since this is a relatively new area, a lot of expertise does not exist in the population in the educational institutes, and they need a lot of training that needs to be provided. And this is what we have started, or the image bank that we are talking of. The logo on the left-hand upper side represents our program, that is the Cancer Imaging Biobank. And this is, as you said, these are the kind of data that we are trying to put into the image bank. We are essentially putting in timeline information, starting from the presentation of the patient, the diagnostic investigation that were done, the radiology, the pathology, the planned treatment that were executed, and the response assessment following that, and any other follow-up up till the point of first progression or death that occurs. In addition to radiology and images, the radiology and pathology images, there are a lot of investigation reports, annotations, and exemplar use case algorithms that we are building using this data, as well as publicly available data that is going to be made available as a part of this effort. So all of this intended, the design of the database, etc., intended from what the data was going to be used for. And this is based on what we are going to do. We were essentially working on these kind of data. There were some tasks which are pure image related, and while some were more complex, we wanted to predict outcomes, response to therapy, and things like that. So the additional factor that went into design of the image bank was the what kind of algorithms that we want to build. Whether we want just black box AI, but obviously the world was moving more towards explainable AI. And explainable AI from the very beginning has to be by design. So starting with the data, there are a lot of additional information that you need to push into the models during training and validation processes. So what kind of intended usage we're talking of? Essentially, simple tasks which are image-based. You don't need anything else apart from image and their labels. Like for the node METs detection, which can also be pan cancer. This is some small work that we did. We showed that most of our algorithms which used our data, we could actually show have a pan cancer detection of metastasis in lymph nodes. There were some cancer types which were particularly difficult, primarily because the morphology of those tumors were very different from those of the other cancer types. And this is where we need a lot of data coming in from multiple cancer types and multiple organ sets into a single database. There were other tasks like simple nucleus detection and classification tasks, and this particular task can actually go on to building novel biomarkers like the stromal TIL scoring that is now published quite a bit, and predicting response to immunotherapy. So the image that you see on the left is actually our own work that we are in the process of detecting and classifying every cell that we see on the whole slide image it's almost big data kind of analytics that is going on. Phenotype-genotype correlation is also a common use case where we want to predict mutations, protein expressions from the H&E image directly. This is particularly useful where the genomic testing introduces a lot of delays in the initiation of treatment, and we very well know that the time to initiation of treatment is a major contributing factor to patient outcomes. Another work where we are actually working on predicting HPV presence in head and neck cancer, and using what we actually saw that the P16IC, which is otherwise a standard part of screening workflow in the Western data sets with high predictive values, actually has very poor predictive value in our Indian patients. So essentially, the difference between the prevalence of the disease and therefore the predictive value of the test is quite low. And this is where we see the use case of putting in AI-based HPV detection detection in head neck cancer and other cancer types. So all of this data is being pushed into the biobank, where we have not just image but also some genomic level information which was routinely done as a part of cancer care. Similarly, work happening with EGFR and EGFR mutation detection in lung cancer. Dr. Rajiv Kumar, the chair of this session, has been contributing actively to this. And one thing that I wanted to point out in this particular slide was actually to show you that how it doesn't work equally. The same algorithm may not work equally in all locations. If you see that that the algorithm that we trained actually performed very well on the same data set.
Swapnil, we lost your audio.
The algorithm accuracies started dropping dramatically, making them practically useless. We have actually found a way around all of this and trying to make algorithms more generalizable as we go ahead. There are other things like response to therapy, outcome prediction, and all of this work can be done on the data that we are providing putting into this image bank. So what kind of data you want to put in? You need to have a structure to the data that is being put in. We have built a lot of minimum data sets based on the clinical pathways that patients follow, and a lot of information starting from the patient level right up to the slide level are being put, including at the whole slide image level, a lot of information, including what tissue is there in the slide, whether it is from primary site, whether it is from the tumor or from the normal area, or lymph nodes, etc., is being put in. Same thing with the radiology images. A lot of information, both patient level, report level, as well as time point level, and these image level information are being put into the data sets. When we started designing and planning for the image bank, we had this big issue of how should we go about all of this? And this is something that everybody commonly faces, whether we go prospective or retrospective. Prospective has each of the ways has their own advantage. We actually went ahead with retrospective because we could actually overcome some of the limitations of the retrospective approach by harnessing prospective clinical trials that were completed in our organizations. So this was something that we found a middle ground where we could take in some of the advantages of the clinical trials and harness them into a retrospective cohort. So we went ahead with cohort and clinical trial driven data. We had a set of stringent inclusion-exclusion criteria as far as follow-ups, as well as the kind of data that is available. And we focused on clinical pathways. So no cancer type is a single disease. Patients get treated in multiple different ways. We wanted to make sure that the AI actually focuses on the cancer and not on the other allied issues, other human-introduced variables into the picture. For example, treatment modality form an important element as a confounding variable in the outcome prediction. So we actually focused on treatment pathways and made sure that we have clinical trials that focused on each treatment pathway and took data from it. Obviously, it is impossible for a single organization or a small set of organizations to have all the data and all different types of clinical trials. And one of the problems that we faced was in an Indian population, certain treatment pathways are the least common. So, for example, we have a heavy head neck cancer workload, but most patients that we deal with, most trials that happen, actually deal with either operable or locally advanced cancers. And on the other hand, the treatment pathways in lung cancer, most of our patients are stage 4 disease at presentation. Therefore, we hardly have any operable lung cancer patients data in India. Data anonymization, protection of patients is an important factor to consider during image banking. Removal of patient health identifiers is obviously the most important, but at the same time, it is important that we don't lose the connection between the data points, maintain the linkages between the different data that is there. TCGA does this through its own IDs, and we have been inspired by that, and we have built our own ID structure to actually recapitulate the maintain the linkages between the data types, at the same time making data access easy. A lot of the slides and etc. can be filtered straight away by simply filtering through the ID fields. Apart from the data from patients, we obviously need a lot of annotations. Finding experts who actually put in the time to annotate is obviously a difficult task. This must be the experience for most people. And another thing is that there's a lot of inter-observer variation, and therefore the usability of single export annotations in AI models is of question. So what we actually harnessed was our strengths, that we had numbers. We have 9 organizations spread out across the country. This is a reporting platform, and what happens is this is the voluntary participation of pathologists in the entire annotation process. And when the pathologist tries to log in into the reporting platform, they get, instead of a Google reCAPTCHA, they get a pathology reCAPTCHA, where images are being present, pathology images fetched from the whole slide images are being presented to them with the binary question, binary labels being assigned. This particular data is actually sourced from multiple pathologists across our country, and then is converted into annotations which have inbuilt measure of confidence in the annotation at the patch level. Further downstream, there is an algorithm that are trained on this data, and as you can see that the algorithms actually we found were quite robust, and they could work on multiple different cancer type, multiple different data set origins, including those from the TCGA as well as our own in-house algorithm, in-house data. Another technical problem that everybody would face is the proprietary file formats. Most of the software solutions are focused on a single file format. A pathology DICOM is still in the making and is a welcome change that will happen. But till then, you obviously need a vendor-neutral image management systems that can harness, that can access all of these images. But in addition to that, they allow open data access, have APIs for fetching individual patches from within the WSI, as well as managing the annotations and perform other administrative and backend tasks. The image biobank that we have established is actually powered by Automation. A big shout out to call out for them. This is a Belgium-based company which has extremely strong image management systems that they have built. The other thing that we focus on is the quality of the data. The whole slide image quality is affected by a number of factors, including the scanner and as well as the slide quality, in the tissue, the artifacts of processing, staining, cutting, etc. We can obviously do a manual quality control, but we are focusing on doing automated quality control using algorithms which are either publicly available or we have developed in-house. So Histo-ROI is one such example, which is a algorithm developed in IIT Bombay and Tata Memorial Center, and we are putting in the results of these algorithms also as a part of the image bank. We do have a lot of checks to make sure that the minimum data set, the mandatory fields are present everywhere. Finding human resources that are willing to work, especially from the IT side, was a big issue for us. We built some private public partnerships as a part of the process, which helped us overcome a lot of these problems. And we had to strike a balance between how much detail we want to collect versus ease of data entry. We had to create a lot of automated checks, validations, and upload procedures that made data entry easy for the medical staff as well as the support staff. Data access policies are still being worked on. We are still working on whether to open up the data for research use only or whether to allow commercial use, and whether what happens if data is been accessed from within India versus outside India. And a lot of these policies would be governed by the policies of the Department of Biotechnology and the Government of India, which is going ahead with the data protection laws of its own. Another regulatory issues making the data open publicly is the data protection laws are different in different countries, and it requires a lot of more concerted effort to make sure that the data in the bank is in line with all of those organizations. So this is a snapshot of the current data that it is available. It is currently still not publicly available, but that is the intention, and hopefully in a year's time, the first data sets would be available publicly. We have begun our work with head, neck, and lung cancer, but we are putting in other cancer types, and there's going to be an expansion of the project to include other cancer types and other organizations as a part of the contributors. Thanks a lot. I would once again take this opportunity to thank my collaborators, the organizing team, and everybody for listening and giving their time.
Thanks a lot, Swapnil, for the excellent overview about the journey of the biobank, which we have started in Tata Memorial Hospital along with the other collaborators, especially from India, and it will open a wide perspective for and you will now shortly receive many calls or the messages from other collaborators or the request for the data, because there is a real scarcity of good quality data which is anonymized and available to the other people. So